Hi everyone, I am Naveen Kumar, graduate student from National Institute of Immunology in New Delhi. And today's presentation, I'm presenting this talk titled "The Non-canonical NF-κB signaling promotes gut inflammation by restraining the tolerogenic beta-catenin LDH2 axis in dendritic cells." So the non-canonical NF-κB pathway is activated by receptors such as lymphotoxin beta receptor and GM-CSF receptor, which activate the SNCC-IKK1 kinase complex, eventually leading to the nuclear translocation of this LB52 heterodimer, which activates the genes involved in immune differentiation and lymphoid organogenesis. So this pathway, the components of this pathway, the receptor and the signal transducers, have been linked to risk loci in human IBD, and this pathway is frequently found activated in biopsies from human colonic biopsies from human IBD patients. So we analyzed the publicly available single cell data derived from mouse gut, and we found that mononuclear phagocytes, which include cells like dendritic cells and macrophage, they express the receptor for activating this pathway, and they are one of the main recipient of this non-canonical NF-κB activating signals like lymphotoxin beta and GM-CSF. We also analyzed some publicly available single cell data from colonic biopsies of human IBD patients, and we could see that the expression of these non-canonical NF-κB transducers, LB and NF-κB2, they were increased in case of colonic biopsies from IBD patients in DCs. And also in the same colonic biopsy samples from IBD patients, the expression of these non-canonical NF-κB target genes in DC were also increased. Indicating that this heightened non-canonical NF-κB signaling is associated with human IBD. We also analyzed some publicly available single cell data set from DSS-induced colitis models of mice. So DSS is a reagent, is a chemical given to induce gut inflammation in mice. So we see that as in the course of DSS treatment, the NF-κB2 and LB expression levels were increased in the DC from the gut of these colitogenic mice, and also the target genes of this non-canonical NF-κB pathway in DCs were increased in the course of DSS treatment. We also did our own studies where we treated mice with DSS for 5 days and then purified ML and DCs, and found that the level of non-canonical signaling were increased in these DSS treated mice. So this led us to ask the question: does this LB NF-κB signaling DC has any effect on DC function in the context of these gut inflammatory disorders, inflammatory gut disorders? So to address this, we generated a mouse genotype that lacked this LB specifically in the dendritic cells, and subjected those mice to DSS-induced colitis by administering DSS in the drinking water. So interestingly, the DC-specific LB knockout mice, they were resistant to DSS-induced colitis as compared to the wild-type corresponding control wild-type mice. As you can see from the less body weight decrease in these knockout mice on DSS administration. This was also shown in less colon shrinkage in the knockout mice on DSS treatment, and decreased inflammatory gene expression in these knockout mice. So we, since these mice were resistant to DSS-induced colitis, we asked what could be so important in these LB null DCs in these mice. So we analyzed some. We did a pathway enrichment analysis on RNA-seq data from the NF-κB2 deficient BMDCs, and we found that in these non-canonical NF-κB2 deficient BMDCs, there was an enrichment of retinol metabolism pathway. Going back to literature, found that this pathway was particularly important to these DCs, wherein gut DCs express this enzyme called RELDH2 that convert retinol to retinic acid, which is a tolerogenic gut molecule. often leading to accumulation of FOXP3, tolerogenic FOXP3 positive regulatory T cells. When we did qRT-PCR analysis, we found that in the renal disease, there was increased expression of this LDH2 basally as well as after LPS treatment. We also did activity assay where we see that in the RLB deficient disease, there was increased RLDH activity. So overall, the non-canonical NF-κB deficiency leads to increment of this tolerogenic LDH activity in disease. Given that the retinoic acid has some role in accumulation of Tregs, we thought to analyze the T cell compartment of these mice. Interestingly, what we find is in the knockout mice compared to the wild type, there is an increased accumulation of FOXP3 positive T regulatory cells, both in the lamina propria as well as in the MLN of the gut. And also, this was true in the after DSS administration also, where we still see an increased accumulation, buildup of FOXP3 positive T regulatory cells. These FOXP3 positive T regulatory cells from these knockout mice showed increased expression of CCR9, which is the gut homing receptor, overall indicating that this disease-specific non-canonical NF-κB deficiency leads to more and more accumulation of Tregs in the gut, giving the mice overall tolerogenic phenotype. Since this retinoic acid is also known to, DC-secreted retinoic acid is also known to, say, promote IgA-positive plasma cells, we measured the IgA-positive plasma cells and found that in the knockout, these IgA-secreting plasma cells were more compared to the wild type. This also coincided with increment in the fecal IgA levels in these DC-specific RLB knockout mice. Again, given that this IgA is known to modulate gut microbiome, we did a 16S rDNA sequencing in this wild type and knockout mice. On some basic analysis, we found that firmicutes, a gut beneficial bacterial phyla, were increased while another gut bacterial phyla, actinobacteria, was decreased in the knockout. When we did some further analysis, we could not see any significant difference among various bacterial genera in the knockout compared to wild type. But to our surprise, we purified the IgA-coated fraction of the fecal gut microbiome. We could see that some of the bacterial genera were altered in the knockout as compared to the wild type. So again, this could have some, this fecal IgE, which was altered due to RLB deficiency in disease, could have something to do in modulating gut microbiome. So since all these effects are attributed to LDH activity of the disease, we thought to ask how RLB can regulate the LDH2 activity. We went back to the literature and we found some factors that are known to regulate LDH activity and the LDH2 expression. So we found that one of those factors is beta-catenin, and it is very highly expressed in these RLB deficient dendritic cells. We also analyzed the disease from the mesenteric lymph nodes and found that indeed there was an accumulation of beta-catenin in the disease from the knockout mice, the RLB null disease. So we wanted to ask how in RLB null there could be high beta-catenin. To give you a brief overview, in the steady state, this beta-catenin is constitutively degraded by this destruction complex. On our analysis, we found that one of the components of destruction complex, exin-1, it was expressed less in the knockout as compared to wild type. Also, the protein levels were low. And we did our further analysis. We found that in the interaction studies, there was a reduced accumulation of this exin-1 to the beta-catenin in the RLB null disease. Going further, when we did a ChIP-seq analysis, when we did a RLB ChIP-seq in the wild type BM disease, we found that RLB was able to bind in the promoter as well as in the intronic regions of this exin-1. And both of these ChIP-seq peaks coincided with kappa-B sites, where RELB could be RELB were able to bind. So this overall these experiments indicated that this RELB could control exon 1 transcriptionally and thereby could control beta-catenin, which can have an effect on RELDH2. But prove it conclusively that and the non-canonical RELB is modulating beta-cat and thereby RELDH2. We decided to generate yet another knockout where there was RELB deficiency along with beta-catenin haploinsufficiency in DCs. So we analyzed the T cell regulatory T cell compartment in the gut from these mice. As we see in the knockout, there is accumulation of FOXP3 positive T regulatory cells from the as compared to wild type. Interestingly, these FOXP3 positive T regulatory cells, they fell when there was a beta-catenin haploinsufficiency along with RELB deficiency. The same was seen in case of fecal Ig levels, where we could see a fall in the fecal Ig levels in the RELB deficient beta-cat haploinsufficient mice as compared to the RELB deficient mice alone. These phenomena also were in line with DSS-induced colitis score, where we could see the colitis phenotype was reverting in the RELB deficient beta-catenin haploinsufficient mice, much like the wild type mice. So overall, this set of experiments indicate that it is beta-cat who determine the immunoregulatory DC function of the non-canonical NF-κB pathway. So this is our model till now, where we see the non-canonical NF-κB pathway activation in DCs leads to limits beta-catenin, which in turn promotes RLDH2. So and to maintain overall tolerogenic gut environment to fight off, say, inflammation. While if there is a deficiency of this non-canonical RLB-NF-κB pathway, there is more of beta, more of beta-cat, and the mice is protected from colitis. I acknowledge Dr. Somen Basak, my principal investigator, and my lab members, especially Elvina Deka, and the collaborators which helped in various experiments, and also thank you BSI for this opportunity to present this study at this platform. Thank you very much.
Thank you, Naveen. There is a question in the chat. How does beta-catenin affect any other immune regulatory pathways?
It's an interesting question. I'm not sure about any immune regulatory pathways, but beta-cat does affect, and not in case of DC, but say high levels of beta-cat does affect with cellular proliferation. In case of DC, I'm not very sure, but there was one paper where the DCs which were, say, had more, they had a larger lifespan, they were more tolerogenic. So I'm not exactly sure how beta-cat can affect the immune regulatory pathways as such. Yeah.
There's also another question from Dr. Zarin. Can you comment on the non-canonical NF-κB in the context of immune tolerance? How is this pathway differentially induced in TREGs, for example, and does it impact cell survival or repair?
Yeah, the canonical NF-κB pathway in T cells does play a role in maintaining their development and functioning, that I know. There has been a knockout of this P65 and CREL in T regulatory cells specifically, and they have shown that those T regulatory cells were not functional. That there has been a study, and there has been studies of that kind. Yeah.